Today I will talk about new solutions, viral and non-viral platforms that we designed for T-cell engineering and really help the field of cell and gene therapy. And really the situations that we're facing right now is we see a new therapy being developed and using cell or genes as a therapy. And we see this, the demand is growing, the results have been really phenomenal, and now there's really need to better manufacture those products. And at Thermo Fisher, we're really committed to provide the best platform solutions to enable cell and gene therapy. Now, one of the issues, the current commercial manufacturing process for viral vector productions is actually unfit to meet the growing market demands. And it's really around the scale and the volumes that we need to develop for cell and gene therapy. We need to make sure the processes must maintain biological activity, answer stable transgene expressions, and prevent transmission of infectious disease while complying with strict regulatory guidelines. And so the challenge is really around design platform technology that can enable efficient and inexpensive upstream and downstream process. And inexpensive is a key word here. We want to make solutions that are cost efficient so that those therapies can actually be applied to many patients, and that's really our commitment. Now, this webinar is really focused around T-cell therapy specifically, and this slides really kind of show the complexity of manufacturing T-cell therapy. It's not as easy as the small molecules or recently we've seen antibodies. There's a quite more complex system, and right now still done at fairly small scales. And really the challenge in the next few years is how can we scale this up? How can we manufacture T-cells in a robust fashion so they can actually be used widely as a therapy? And the process, if you look at T-cell therapy, is first you need, right now, is a lot of autologous T-cell therapy. So you need to get the T-cells from the patients. There's a selection process to select the T-cells, to separate the T-cells from the B-cells and other cells in the blood. And then there's a big engineering part in the middle when you want to genetically modify those T-cells so they can actually specifically express a receptor that can then recognize the cancer cells, which is the key aspect of this therapy. You then need to expand those T-cells in large amounts and then finally infuse them back in the patients. So you can see there's a lot of processes and manipulations of the cells that can be done right now at the lab scale, but really we want to kind of be able to scale this up. This webinar is really focused in the middle here, which is what are the ways to better activate a gene transfer and to better engineer those T-cells. So I'll focus specifically on Lentiviral Production System that we developed, but also the new generations that we see is coming, which is Electroporation device as well as gene editing. Now, we also have a lot of product in the other area, isolations and beads, and I'll talk a little bit more about this in the next slides. So now that's another way to look at this workflow. You see, we look at cell isolation and activations, cell engineering, like I just talked about, cell expansions, where there's a lot of needs for new media, serum replacements, growth factors, and as well as single-use technology. Wash, fill, finish, and cryo. This is also a very important area. You want to make sure those cells are sterile and clean and produced under GMP conditions so they can be safely injected in patients. And then lot release and characterization. There's a lot of characterizations and QC all along this process to really make sure that the cells you're trying to inject are pure, they don't contain contaminants, and they're ready to use. And so there's a lot of safety, identity, functional analysis, cellular analysis, protein, and genetic. Now, within Thermo Fisher, we have a lot of those products already, and I'm not going to list them all, but kind of give you an idea of some of the key products that we have in this area. For example, cell isolation and activations. We have the Dynabeads that are produced by our colleague in Dynal up in Norway, and those beads are actively used to actually either isolate the T cells, but also to grow and expand the T cells. And actually, some of those beads are already used in a clinic to prepare those T cells. In the cell engineering, this is where I focus mostly, so I'll skip this. Cell expansions, we have media that allow you to grow your immune cells and T cells specifically, such as the M5 media, optimizer, and also we do a lot of custom media to kind of, depending on your needs, we can specifically formulate a media for your needs. Optimizer and are used right now in the, to expand T cells. We also have human serum replacements, which allow you to get away from using human serum, which can be costly, not very robust, a lot of variability. So we, get rid of this variability by having a more defined serum replacement, and that can also work. We're also developing growth factors, CTS. What we call CTS is really a cell therapy system. So those products have the highest quality standards, and not only GMP, but they also have extra quality to really maintain the best quality possible for T cell therapy. We have a lot of different growth factors, such as interleukin 2, 4, and 7. We also have a lot of single-use technology. So those are the bags, the bioreactors that you can actually use to grow your cells. So we have bioreactors. We also have rocker reactors that allow you to grow your cells. And watch-fill cryo, we have products such as CTS, PBS, but also cryopreservation media. And then on the characterization and safety, we have a lot of molecular tools that can be used for this, such as qPCR or next-gen sequencing with the Iron Torrent platform, or flow cytometer with Attune and XT. We have a lot of product and solutions, as well as a lot of antibodies to do a lot of characterization. So really kind of overall, set of tools to be able to do those gene-modified T cells workflow. But really, like I said, the focus is around cell engineering, and specifically to start with, really the lentiviral production system. So T cells are very hard to transfect, and you can't simply use the transfection reagents. And you have to use a virus. And in this case, lentivirus is kind of the preferred method to transform those T cells. Because not only you need to introduce the DNA inside the cell, but you also need that DNA to get introduced into the genome of the T cells. So those T cells can express for a long time the chimeric antigen receptor that you're trying to express at the surface. Because what's important with those therapy is once you inject those modified T cells into a patient, you want those T cells to stick around for a long time so they can actually do their job, which is homing onto the cancer cells, kill the cancer cells, but also remain in the body. So if any new cancer cell appear, they can actually get rid of it. So it's important to have a sustained expression of this receptor. And lentiviruses are really perfect for this because they incorporate the DNA inside the genome. Now, making lentiviruses is not easy. As you can see on this slide on the left side, what you need to do is you need to use a packaging cell lines. So typically those cells are 203 cells, HEK 203 cells. And you need to transfect into them different vectors. One vectors contain your gene of interest, which is on the left, whatever gene you want to incorporate. And the three vectors we have on the right are the packaging vectors. So you need to do a transfection of 4 different plasmid DNA, transfect into those 203 cells, then the lentivirus will be released by the cells in the media, and then you need to harvest and purify it and get those LV. Now, if you think about it, you injecting millions and billions of T cells, and per each T cells, you need at least 2 or 10 lentiviruses to be able to transduce those T cells. So you need a lot of lentiviruses. Now, one of the challenges, a lot of the system on the market right now are using adherent system, meaning the cells, the packaging cells, 203 cells, are sitting on the bottom of the dish, and you transfect them. As you can see on the upper right, you use a lot of Petri dish to do this and changing the media. Works great for the small scale, the research scale, but if you try to scale this up, it becomes a challenge because you need this adherent surface, and we have like cell factories that are stackable, large plates that you can grow a lot of cells at the bottom. And if you want to go even further, now we have walk-in incubators and really kind of those giant T cell factories that you can use for that, the plate factory. But it's a problem. You can imagine First, they're hard to scale because there's a lot of plastic involved and there's a lot of manipulations that can be really heavy. And also, some of the system contains serum and really overall, and then low titers. So because of the low titer, they're hard to scale and they become very expensive. And you know, in the whole T-cell therapy workflow, they can represent 40% of your costs, and that's a big problem. And really the goal that we want to do in the system is really reduce the cost of lentiviral productions to really help and fulfill the needs of these markets. Now, so here I'm talking about the new product that we launched, which is called LV-MAX. We're not really new in this business. There's some of our products are already being used on the market, such as lipofectamine 2000 CD, for example, which is used as a transfection reagent to make lentiviruses. So some company already using it, and they are using this product to make large-scale, commercial-grade LV productions that are actually used in the clinics. And so, if you look at lentiviral production system, here this last kind of there's a first part, which is the vector constructions. You know, you want to clone your gene and do all kind of molecular, put different promoters or kill switch, etc. So for this, we really use the power of our one of our group in Germany called GeneArt, which are a gene synthesis company, and they can fulfill all your cloning needs and allow you to clone your vectors. In terms of vector productions, like I said, there's a lot of people using still adherent system right now. But really our goal is to move to a suspension system that allow you to get more scalable system and really overall reduce the cost. And I won't talk about it today, but we also downstream of this, we have another group that's focusing on vector purifications. And you may have heard, brand like Poros, Capture Selects, affinity matrices, and what those guys are doing, they actually develop affinity matrices to purify viruses. And purification can definitely be a challenge. You can lose up to 50% of your lentivirus through the purification process. And we really try to improve this with more smarter system affinity matrices that allow you to kind of better isolate your viruses and scale it up. So right now we have matrices for AAV 8 and 9, and this group is developing new matrices for lentiviruses. So if you're interested, please contact us, and we can definitely put you in contact with this group. But let me focus on the middle here, which is really around vector productions. And I'll say a few words about the adherent system. So like I said, there's a lot of adherent system being used right now. And so we thought a lot of other systems actually use older generation transfection systems, such as calcium phosphate or PEI, that work to some degree. But what we wanted to show here in this work is showing that by simply switching the transfection reagents to 2000 or even to our best transfection, which is lipofectamine 3000, you could achieve really higher yield and allowing you to get a lot better titers and overall obviously lower the cost. And this is slide kind of showing this, some examples when we use 2000 and 3000 and you see a lot better yield. So yes, the transfection reagent may be a little bit more expensive than PI to start with, but because you get such better titers, the overall cost of your LV is actually will be lower. So if you're interested, if you don't want to move your system, if you want to stay in an adherent system, we simply suggest that you may want to try to swap just the transfection reagents and you'll see better titers. We have an app note on our website about this, kind of detailing the protocol on how to do this. But really the focus here is we wanted to radically change the process, and most importantly, we wanted to adapt the process to be in suspension. Suspensions allow you to really scale this up and kind of overall reduce the cost of the manufacturing. So to reduce the cost, though, you need to have really high titers. So we wanted to develop a system that does not lose the titer, actually even better than adherent. In general, adherent cells are easier to transfect, and you tend to see pretty good titers. And once you move to suspensions, because the cells are in suspension, they have a lower cell surface, membrane surface, and they'll be hard to transfect, and then you can lose some titers. Our requirement was really to get titers at least equivalent to adherent and even better. Scalable, obviously, because the suspensions, you can now take them from a shaker flask to bioreactors. And ultimately, we want all of our product to be GMP, media, cell, etc., so they can actually be used to the clinic. So let me say a few words about this. So the way we attack this system, this problem, is we look at the system approach. A lot of people trying to optimize the lentiviral productions, they buy cells from different companies, different media, different transfections, and kind of try to figure it out, put this together. Well, we pretty much did the same, but at a large scale, meaning we wanted to optimize and find the best cells for these applications. We wanted to screen and optimize the best transfection reagent for these applications. We have a lot of experience through the Invitrogen days in life technology. We have a lot of experience in transfection reagents since 1987. So we have a lot of reagents that can be specifically designed for these applications. We also wanted to find new enhancers and new supplements. And so you can see there's a lot of different factors from cells, transfections, media, supplements. And so we use here design of experiments to really optimize each component. So not only identify the best cells, but also make it work together with the other components. And for that, design of experiment was very useful. The goal was to get a system that is a high titer, very high titer, more than 110⁸ TU per ml, and this is unconcentrated. And the titers is functional titers in which we transduce cells and count the transduced cells, so it's not qPCR. Scalable, that's the key factors, and then GMP, so they can actually be used in the clinic. So let me show you our approach, what we've done in the lab to really design these products that we call GIBCO LV-MAX. And kind of showing what we've done in the lab here. So you can see on the left, we needed to screen a lot of different reagents, different cell lines, different transfections, different media and supplements. So, and all in combinations at different ratios. So there's a lot of experiments to be done here. So really, to be able to do a massive screening of all those components, we use 96 deep well plates. So that's the beauty about suspension cells. You can actually use deep wells and check the cells in each well, and they'll allow you to do a lot more combinations. And then once we find the leads and new products, we then verify it into shaker flask, first moving at 30 mils lenti productions, then to 1 liters, and now we're in the process of verifying the protocol and really adapting to larger platforms such as the Wave or bioreactors. And like I say, all our titers were functional on HG1080 cells. For the screening, we'll use a lot of GFP as a readout vectors and flow cytometry to really look at the titers. So now like I said, this is a system. So LV-MAX contains cells, media, supplements, enhancer, and transfection reagents. So all you need to produce the LV. What I want to show you kind of the next few slides in each component of this kit. So first, we have the cells. Those are cells, like I said, the 293 derived. They can grow in very high density and stay healthy for a long time. So it's important, and in suspensions. So those cells you can actually achieve up to 13 or 14 million cells per ml. So very, very high density without losing viability, and that's very important. Also, those cells, do not aggregate or clog, which make it very adapted for developing high-density system like this. And this is showing here the growth curve with the viability in the upper left, and you can see those cells can grow very fast and very healthy in a suspension environment. So you have the cells. Now, something that is underlying everything is not only isolating the right cells, but also the right protocol. For example, even though those cells can be grown in 12 million per ml, you can imagine that high density will achieve the transfection, will affect the transfection efficiency. So you need to really find the right balance of the right density to have the maximum productions, but also the right density to have a maximum transfection. You can have 12 million per ml, but if they can't transfect, then you're going to have actually overall less. You're going to end up having less lentiviruses than if you start with lower concentration. So I won't go through all these slides, but kind of showing all the optimization that we've done and working on the protocol to really allowing our customers to have the same result that we see in the lab. And so there's a lot of work being done on the protocol level to really kind of optimize each parameters and guarantee robustness in the system. Now, the other thing we looked at is the supplements media. And the supplements is a system that we use to allow us to control the growth of those cells. The last thing you want is have the cells to grow too fast, because when they grow too fast, they actually spend a lot of time multiplying. What you want to do is really slow those cells down so they can actually spend more time producing what you want them to produce. And it's a phenomenon that we use a lot for protein productions. It's kind of similar for lentiviral productions. When you want to slow down the cells in such a way so they can actually maximize productions. If you slow them down too much, then you slow down the machinery too much, and therefore they won't produce enough viruses. And here the slides show this on the upper left. We use different concentration of the supplements, 1, 5, and 10%. So you can see by this, we can achieve the cell growth. And if you correlate the cell growth with the titer production, which is at the bottom left, you see that we can find optimum concentrations to really maximize lentiviral production. Now, notice that because it's multifactorial, there's a lot of components coming into play, and this is where design of experiment is very useful. Notice this experiment is not as simple as using supplement at different concentrations. There's also cell density being involved, as well as the amount of DNA and the transfection reagents, and as well as the passage of the cell. So there's a lot of multifactorial variables that you need to consider and look into in the context of all those different reagents. Now, so cells supplement to slow the cells, but also transfection is very important. And in the past, my group, we've been developing transfection reagents. We try to develop transfection reagent that works on many different cell lines with many payloads, kind of have a broad spectrum reagents. In this case, though, we can be a little bit more specific because we go specifically after one cell line. We can control the conditions better, and we know we need to transfect for plasmid. So transfecting cells in suspensions is typically more difficult than adherent cells because, like I said before, the membrane surface is different. But also the density can affect, and then also transfecting 4 different plasmids can also be a challenge. So here we went back into our library of reagents. We have accumulated a lot of different components, compounds over the years, and really screened specifically for these applications, where we went through a lot of different reagents, translating 4 different plasmids into the cell lines, and really tried to identify the best transfection reagent possible. And here, this is an example of a DOE run, and you could see different code name for our different transfection reagents, and you could see we isolated quite a bit of transfection reagent that were performing better. Now, obviously, this is what we do in the lab, the screens that we do. For the kit, we only pick one, put in the kit, and recommend this one. And so, like I said before, there's also a lot of underlying work to optimize the protocol. And here showing kind of a little bit of a sample of the type of things we do in the lab. So not only we need to select the best transfections, but once we selected the transfection, we need to optimize the protocol. And there's factors that are important, such as diluting the transfection reagents in the optimum. The time of incubations can have drastic effect on transfection efficiency. And you can see here on the upper left, if you transfect if you dilute into optimums, you have about 1 to 2 minutes to then proceed with complexation with DNA, because you will quickly lose activity if you leave it too long in optimum. So really, incubation in optimum is critical for the reagents. For DNA on the upper right, it's less important. You can see the DNA is a little bit more stable in optimum, so you have a little bit more time. Other additions also matters. Although in this case it doesn't matter too much, but we're also looking at this as well as the complexation mix and the time you can incubate and the stability of this complex. So I don't want to spend too much time on those graphs and the details, but what I want to highlight is I we try to simplify a protocol and give you guidelines on how to do it, but you can see behind those protocols that may look simple, there's actually quite a bit of work that were done in the back to really kind of optimize the conditions. And not only making for the best performance, but also the most robust way, so everybody can have the same result that we get in the lab. Another aspect, reagents we went after to improve LV production is the enhancers. There's enhancers you can use that allow you to actually, the cells to release more lentiviruses in the media and to improve the budding out of lentiviruses. And here again, there's a lot of different components you can look, and this is where design of the experiment is very useful. But here, this is a result of a screen. You can see, again, all kind of different candidates here compared to a control, which would be no enhancers. You can see we found a lot of different enhancers that can drastically improve the titers of those lentiviruses. And you can see we can easily achieve, 1.5 to 10 to the 8 lentivirus per ml. Again, this is not undiluted, so I get a lot of questions is, if those lentivirus are concentrated or not. They are not. We just simply take lentivirus from the media, we do serial dilutions and transduce HT1080 cells and then measure the titers by flow cytometry. So you can see a lot of improvements there on terms of titers and coming from the enhancers. So again, we do a screen in the lab and then we pick one that we put in a kit. I'll skip a little bit on this one, but again, you can see the time of additions can matter. We tested, whether you put at the same time you do the transfections or 4 hours later, 6 hours, 8 hours. Now, the nice thing is you actually have a longer window between 4 and 16 hours. You need to add the enhancers to have the right activity. And that's kind of important for the protocol, which is on this slide here, where we want to give you really the best protocol, most robust to achieve your performance. So we recommend to see the cells at 3 million per ml. Now, one issue that we've noticed here is people count the cells differently. There are different machines that can give different counts, so sometimes there's more variability there, so it's important to kind of be consistent there. So if you don't see the exact result that we see, you may want to kind of change a little bit of the density around 2 to 4, depending of the way of counting. Then you would simply add the supplement at the time of the transfection, so you slow down your cells. Then you prepare the complex, you do the transfection reagent, use the transfection reagent, and you prepare the complex. You add 10 minutes to the dilute cell, 10 minutes to the diluted cells, and then post-transfections you add the enhancers, and then you could harvest 48 to 55 hours. Now, these slides kind of show the progress that we've done through LV-MAX. You could see prototype 0 is kind of where we started, where we took reagents from, we did what a lot of people do. We took different reagents commercially available, assembled them, and see what we get. And you can see that's where we started prototype 0. And through optimization of each component and working the protocols together, we can now achieve, the LV-MAX reagents results, as you can see on the right, achieving in this case 5 to 6, 10 to the 8. This is done in 96 well plates. We tend to see better titers in 96 well plates. We're not sure why, but probably because of the mixing. But we definitely achieve at least 1 to 2, 10 to the 8. But you could see a really nice improvement through optimization of each reagent and working them together. Now, this slide 22, really highlight the importance of a system, because it's not just about the cell line, it's not just about the transfections, it's about the combination of all those things and how they work together. So yes, we selected the best cell lines that we could, the best transfections, the best enhancer, and the best supplements, but making them work together is actually part of the major optimization and development that we've done. And here showing, in this swapping experiment, we're showing that if you change LV-293 with 293 freestyle, for example, you can see you're going to lose quite a bit of activity of lentiviruses. Transfection reagents, I was here lipofectamine reagents are more expensive than PI, which is true, they are. But it's really not about the cost of the reagents, the cost of the lentiviral production. So yes, you may use a cheaper transfection reagents, but what's going to happen is you're going to have a lot less titers. And if you look at the price per titer, then you are going to end up spending a lot more money to produce LV. And I'll show you a little bit more on the next slides. Supplements are very important, enhancers are critical, and really kind of show that if you use them all together in a protocol, then this is where you can achieve the best results. Now, it's scalable, and that's the key really aspect of this because it's suspensions. You can actually do 96 deep well plates, which is great if you want to do a lot of lentiviruses and screen and find out which one is the best, which CAR is the best for your T cells, and you could do a lot of research this way. But you could also take this product all the way up to the chain and moving to bioreactors. So here showing different shaker flask, but also now we're working with our colleague in Finesse to really develop 2 liters and even plus, bioreactor protocol to really kind of take it to the next level and be able to do 200 to 500 liters scale. And this is important. So in this slide, we really wanted a product that you can use from your R&D to the clinic. So you don't have to change your protocol along the way. You don't have to change the reagents, and that may ultimately affect the performance. You can actually, do small scales, large throughput, non-GMP open platforms. You can do a lot of lentiviruses, and then slowly move up to the clinical aspect. So preclinical, you can do medium scales, and then clinical bioreactors. And so ultimately, we want a kit that can do large scales, low throughput, and GMP, and in bioreactors. I should mention that the kit right now, Ginkgo LV-MAX, some components are GMP, but not all of them. And we're working towards making all the components GMP. And this should be some components will be available sooner than others, but ultimately the whole kit will be available next year as a full GMP version. This slide kind of highlighting the price. Like I said before, we hear that PI is a cheaper reagent as transfections, which is true, but because you actually get typically 10 to 15 times less virus, the cost of LV ends up being 52, 58% more. And it's easy to imagine, especially if you move to different scales, if you need a certain number of LV particles for your needs, let's say if you use PI, you will need, for example, a 10-liter batch, but if you use LV-MAX system, you will only need a 1-liter batch. So you can imagine there the price difference in terms of media and cells and cost and people and time that really at the end of the day you save money. And this is really the goal of this product. The goal of this product was to reduce the cost of manufacturing lentiviruses, so actually everybody can use this therapy and develop T-cell therapy. So this is the kit. Like I said, it's a system that has media supplement, high density, new transfection, and enhancer all together and really worked out together. This product has been launched in July 2017, so it's available now. We have different formats, and really it's the first optimized lentiviral production system. So we've done the work for you. We've selected the best reagents for each and then put them together. And like I said before, it's animal origin free, scalable, serum-free, and we get really high titers. And the GMP is coming. The full version of this kit will come next year. But if you're interested, we have media, for example, that are already GMP. We have different configurations. We have the standalone that comes with the cells and the media. So if you need to order more media or more cells, we have the starter kits. This is what we recommend to start, which is really, has everything you need: the cells, the media, the transfection reagent, supplement, and insert. And then separately, we offer also the transfection kit that has the transfection reagent, the supplement, and insert separately. So this is the catalog number if you're interested, but really kind of go on our website, thermofisher.com/lentiviral, and you'll have more information here. Now, in the next 10 minutes, what I want to show you is a little bit kind of what we see in the future and what's going to happen. specifically focusing a little bit more on non-viral options. So Lentivirus is really the main platform right now, and it's probably going to be for many years. Works great, deliver T cells. But ultimately what we'd like to develop is a non-viral solutions where you can use electroporations as well as gene editing to allow you to stably incorporate the DNA inside the genome. And really kind of going from lentivirus or / retroviruses to gene editing approaches. One of the limitations of lentivirus, like we mentioned here, is the cost. And we try to reduce this with a better system like LV-MAX. The safety, there's concern about, because the lenti incorporate the DNA inside the genome, there's concern that that DNA can incorporate into existing gene and change the function of the cells. We could have dramatic effects. There's limitation around the insert size. There's a limitation on the size you can put into the lentivirus to incorporate. And as we see, this field is moving toward a little bit more complex system, more subtle regulations of the chimeric antigen, and so that may be a limitation there. Most important, you cannot do gene editing. So, for example, works great for autologous system, but allogenic, you're going to need to do knockouts. And so for that, you're going to need to use a gene editing approach to knock out a TCR receptor, so you can have basically a donor that will be able to give these T cells to patients and kind of develop allogenic. So what we see is really the next generations will be a non-viral approach using gene editing approach that will allow you to do knockouts, knock-in, and really incorporate. Now, what's lacking now is probably around the tools exist, but we want to make them the performance. We will need to improve knockout efficiency, knock-in, and really avoid AAV as a donor DNA. So the question is simple. Lentivirus can transduce cells, T cells, from 20 to 45% insertion. Can we achieve the same with a non-viral approach? And this is really one of our focus as well. And it's really our goal is beat lentiviruses. So we have tools for this. So first is start with the delivery tools. We have a neon electroporation device, which is a device that allows you to simply electroporate cells, and it's very easy to use. You have a pipetter, and the tips is basically the cuvette. So you simply pipette the cells, put in the tips, and then you could control the parameters, such as the pulse width, the pulse size, and the number of pulse to allow you to do transfections. And on the right here, we've done a lot of optimization in primary T cells, where we look at all kind of factors, the donor variability, the activation protocol, the media we use. And what's interesting is if you look on the right, we can achieve 80% transfection efficiency in primary T cells, which is pretty remarkable, knowing those cells are typically very hard to transfect. What's interesting, and we don't fully know why, but you could see at day 3 you get 80%, but day 5, day 7, day 10 through expansions, they become more and more difficult to transfect. So that's something we're actively exploring. The good news is we can already achieve 80% at day 3, which probably will really help our gene editing experiments when we can put DNA at that stage and be able to engineer T cells. Now, the other part that you need is the gene editing tools. And for this, we have a lot of gene editing tools, and basically 2 categories. One are the CRISPR-Cas9 tools that we offer in different formats. I'm sure you all heard about CRISPR. The system that uses a guide RNA as well as a protein to basically go recognize into the genome and cleave the genome. So basically what you need to do gene editing, you need to express the protein Cas9, and you need to have a guide RNA. They find each other, and then they go recognize on the genome and allow a specific, gene-specific, site-specific cut. So there's different way to bring the Cas9 protein inside the cells. You can bring it as a protein itself, which is the RNP format. You could bring it as an mRNA format or DNA format. And, I can talk for a long time about this, but to say simply, we found the protein format is the most effective one. Give you the best results, but also the minimum off-target effects, because the protein is there, do the cleavage, and then quickly disappear. We also have guide RNA and synthetic guide RNA. That's something that the team just came up with. Those guide RNA can be chemically modified to improve stability, but the most important is a single guide RNA. You complex with the protein, and then you deliver. We also just launched a new Cas9 protein we call TrueCut Cas9 that actually give even performance. That's the CRISPR tools. We also have TAL effectors. So those work a little bit differently. They are DNA binding proteins, and there's a specific code, and you could design TAL to target specific sites. And the beauty of this is you design those TALs, and then you could put in fusions, all kind of proteins that allow you to do nick the DNA, or you can add a repressor, activator to turn on the genes. What's nice with the system is you can actually have the two protein. They will recognize the genome, and if you put a restriction enzyme fused to those TALs, once they're recognized, they will work as a dimer and then cleave the DNA. So TAL allow you to actually give more specificity because you must have the binding of the two TALs, and you must have the perfect pairing between the TAL to have cleavage. An important factor too, especially if you want to move commercially with the system, the IP landscape for CRISPR is a little bit more confusing. There's different groups working on Cas9, and it's unclear who really truly own the IP. So if clinically there might be a problem, Talon, we have a clear commercial path. We're only the only provider of Talon technology, and we can actually give you full access to develop. Commercially, it's also give more flexibility. There's less restriction on the sequence you can go after, so kind of give you more flexibility. But again, we have both systems available. What we've been focusing a little bit more is really around RNP Cas9 as well as the guide RNA. And so what we wanted to do is kind of use this tool and optimize. So we have a lot of offering in T-cell isolations and activations. And so this kind of, here's the protocol. We would get T-cells and activate them using adenal beads. We do a transfections with the gene editing tools and the neon transfections. And then we can verify indel and cleavage efficiency using our sequencers or iTunes flow. And here it's kind of showing some experiments that we look at Cas9 RNP delivery in day 3 T cells. You could see on the top of the protocol. We use different sites. We go either after beta-actin or HPRT, just as a control site to verify it. And then here I'm showing a gene cleavage detection assays on the gel, but you could also do next-gen sequencing. But really the goal of this slide is kind of show you this new Cas9 that we call TrueCut Cas9, showing the better performance with this Cas9. And you can see here on the bottom the cleavage efficiency that we already achieved, which is pretty remarkable. With the old Cas9, we were achieving 70%, 60%. Now with the new, we're up to 80%, 90% on those locus. And Neon program mean we use different programs, different pulse or system, but kind of showing the robustness of this platform and really achieving regularly 80 and 90% knockouts on those primary T cells. It's pretty remarkable. On HPRT locus, we see even better improvements with the new Cas9, where the old were giving about 50%, we can now achieve 80%. So you can see we can already achieve in primary T cells with Electroporation platform and our new gene editing tools, we can already achieve really nice knockout efficiency. Here this is another example. Instead of using HPRT and beta-actin, we use TCR receptor, and we design multiple guides, track T1, track T2, TRB, T1 or TRBC T2. You see, depending on the guide, you can have different activity, that's why it's important to really kind of screen and find the best guide RNA. But you could see that in those T cells, with some of the guide, we can achieve 90%. And if you do a functional knockout, so that's 90% of indel efficiency, but if you look at functional knockouts, if you look at the protein itself, we achieve 99, 98%. So really incredible yield, incredible knockouts, and really we don't see this as much as a challenge anymore with the right tools. We can already achieve very important knockouts. So that part is taken care of. So first, you can really efficiently achieve the knockout. Now, really, the next step, if you're going to replace lentivirus, is how do you incorporate the DNA inside the T cells? So not only you need to cleave the DNA, but you need also to have homologous recombination so the DNA can incorporate at the site of cleavage. Now, a lot of the methods right now are can achieve 30 to 40% knock-in. One of the problems, though, is you need to use AAV6 as a donor DNA. So you have a combination of a AAV6 donor DNA with Cas9 gene editing, electroporations, and then altogether you can again achieve knock-in. But it really doesn't fix the problem, because one of the problems of lentivirus is the cost in producing it and you know, the manufacturing workflow. But now you're replacing lenti to AAV6 donors. So it's really kind of, it doesn't fix the problem. What we really want is a whole non-viral approach. And so the question is, can we now replace AAV6 donor DNA with a plasmid and achieve a whole non-viral? And it's already being done. There's actually quite a bit of papers showing that you can use neon. In this paper published by Alexander Martin, you see very nice incorporations of the DNA inside the primary T cells using neon electroporations, about 25%. There's another paper showing, again, same platform, neon gene editing, and it can also achieve pretty high incorporations, knock-in into the HSC cells. So you see this is already working. You could already use non-viral, but those two examples really using small inserts of DNA. Well, for CAR T, you're going to need more than 2 to 3 kb, so it's still kind of a bit of a challenge. So I can't tell you too much, but I can tell we're working actively on this. So we've shown right now that we can achieve donor DNA naked with the Cas9 protein, that, especially the new tools that we have, we could already achieve up to 20–30% knock-in efficiency. So we think we're getting there, and ultimately replace the lentivirus. It's still a long way away, but kind of definitely a work in progress. And if you're interested, please contact us. So I'll stop right here. Thank you for listening. This is really kind of summarizing the cell engineering. We have different platforms. The most ready to use right now and launch is really our Lentiviral Production System called GIBCO LV-MAX. If you want to produce high level of lentiviruses, we strongly recommend this platform. And I'll stop right here, and I'll take any questions. Thank you.